Today we have with us Tom Terwilliger. He's senior scientist in Los Alamos, and he's part of the collaborative project of Phoenix that is led by Paul Adams. And Tom has developed algorithms and software for X-ray diffraction and cryo-EM and many other data analysis of macromolecules and proteins. And I'm sure that we will enjoy with this seminar about AlphaFold. If in any moment you have questions or comments, you can write them in the chat, or alternatively, you can wait until the end and directly using your microphone, you can ask Tom. If no questions, please, Tom.
Thanks very much. I'm really happy to be talking to all of you today. And so, yes, I'm a developer and software writer for Phoenix. And so today I'm going to talk about how AlphaFold and related artificial intelligence methods for high accuracy prediction change everything in structured determination methods, but at the same time, they change nothing. And that paradox is that AlphaFold gives you a bootstrap, a head start that's amazing and fantastic. And it changes everything about how we can start out interpreting maps in crystallography, cryo-EM, and other fields, of course. But nothing changes because these models are not perfect. They still require the experimental data. And so the experiment is just as important as it always was. And at the end of the automatic procedures, we're still left with the same situation that there's a lot of manual work necessary, a lot of comparison with the map. And so nothing in a sense has also changed, but it's very exciting because the bootstrapping, getting started is just fantastic. So here's what I'm going to talk about for the next 35 minutes. First, I'll talk about AlphaFold models. I'm just using them as an example for the set of new high accuracy prediction methods. I'm going to talk about how these models are great hypotheses. And if you don't take anything else away from this talk, remember that they're great hypotheses. They're not the answer, but they're really great ideas. The second thing I want to talk about is that these great hypotheses can really jumpstart structure determination for cryo-EM, X-ray, and other methods. And I'll give you some examples about why that's true. Then third, I'll tell you something you probably don't know, and that is that you can improve the modeling by AlphaFold by implicitly incorporating information from a density map. And that's a new thing, and that's very powerful, as you'll see. Putting all these things together, AlphaFold models really allow a new workflow for structure determination. And I'll talk about that workflow, about how you can think about starting the experiment and how to go through the experiment for cryo-EM and X-ray today, and show you how different it is from it was yesterday. Let me just check here and make sure it's all working and no questions. Jose Luis, can you just verify everything looks good?
Yes, everything is working.
Okay. So let's talk about AlphaFold models as great hypotheses. So let me just review briefly the concepts in high accuracy prediction. I'm not going to tell you anything about the details, but I want to tell you about the concepts that are relevant to how they're useful now in structure determination and how they're useful and not useful. So two aspects of machine learning prediction: first, training, and then prediction. So training involves having a large data set of known answers and a large data set of features that are going to go into the prediction. So the features in this case correspond to a sequence of a macromolecule, a protein in this case, and a multiple sequence alignment that goes with that sequence. We'll talk more about that multiple sequence alignment in a moment. So maybe we have, in addition, a three-dimensional structure that goes with this, and that's the thing that we want to be predicting, and that's the target. So training on some 180,000 structures in the protein structures in the PDB yields some 21 million parameters for AlphaFold. Notice the ratio of parameters to protein structures is actually pretty big. That means that there's a lot of information that is in there, and it's not obvious that pieces of the structure could not be implicitly stored. So there's always a little bit of bias question in this kind of analysis. So now we have these parameters, and you have a particular sequence that you would like to predict a structure for. So you take that sequence and generate a multiple sequence alignment. The multiple sequence alignment is basically an alignment of all the similar sequences that are available in protein sequence databases. And of course, we're using the assumption that similar sequences have similar folds. And we'll talk, as I said, a little bit more about that in a second. Combining that information with all those parameters and focusing attention on important relationships using that multiple sequence alignment that I'll talk about a little bit later allows you a prediction of the structure. And this is a real structure that I'm going to show a few times in this talk. And you'll notice that part of it looks right and part of it doesn't look quite right. Now, in addition to a prediction of the structure, AlphaFold gives you confidence estimates, residue by residue. These turn out to be very useful, and we'll see later how you can use them to prune the bad parts of the structure or the less confident parts of the structure away. I just want to comment that these confidence estimates are pretty good. They're unbiased. On average, they're accurate, but they're not perfect. That is, a high confidence estimate is not always correct, and a low confidence estimate is not always incorrect. Now let's talk about this multiple sequence alignment for a moment. So on the left-hand side of this, I've written a protein sequence. This one's actually made up. And then I've aligned dummy sequences against that, supposedly sequences from the sequence database of actual structures. I've listed in red a few pairs of residues that seem to change at the same time in different sequences. And residues that covary in this way are often close in three-dimensional structure. So AlphaFold and other prediction methods basically have the capability to use the information that pairs of residues that change together are probably close in structure. I note that that actually is not programmed into AlphaFold. AlphaFold learned how to do that. A second thing that one could obtain from a sequence alignment is that all the sequences in the alignment should be compatible with the same correct structure. So for example, if there's a residue that in one sequence would go inside the protein, but it's incompatible with being an internal residue, then it's that structure is probably not right. And AlphaFold can learn that sort of thing as well. Now, the multiple sequence alignment is important in identifying, in generating predictions. And the more sequences that can be aligned correctly, the more accurate the prediction of what residues are close can be done. The lower part of this figure shows a picture of a real structure with the sequence coverage on the left, so number of sequences that are matched to each residue position, and it's pretty high for the part on the left. And on the part on the right of that sequence, it drops to about half. That is reflected in the confidence estimates, or predicted accuracy, which is very high for the most of it, and then less high for part of the right, the part of the sequence on the right. Where the sequence coverage is high, models can be very accurate. So this is that same structure. The pink is the deposited structure in the PDB. The green is the AlphaFold prediction. Notice the left-hand part, which is residues 1 to 100, which had a high sequence covered in that previous slide, has very good confidence and it's very accurate. The right-hand side, which has much lower coverage, is not all that accurately predicted. The part that is accurately predicted can be astonishing. So this is the amazing part of these high confidence prediction methods. It's not that they're like pretty good models. It's they're, in the best parts, they're basically perfect. So here's an example. This is part of residues 1 to 100, and it was showing that the deposited structure and the AlphaFold model, and a lot of the atoms are just smack on, right rotamers, right everything. But not everything. Some parts you can see on the left, there's a couple side chains that aren't quite right, but is just amazing. So high confidence residues can be very accurate. There are some limitations, serious limitations to AlphaFold and related programs. It's only protein for now, so you don't get any water, no ions, no covalent modifications, no carbohydrates, no ligands, no DNA, no RNA. So a lot of structures are missing something. Importantly, AlphaFold in particular has been trained on good and poor structures. It's trained on pretty much everything, all the protein structures in the PDB better than 9 angstroms. So even ones that aren't accurate, even ones that are very low resolution, those are in there. So in the future, that'll probably be fixed. It'll probably be retrained many times by many people. But at the moment, the parameters might systematically include poor geometry. So keep that in mind, that you can't necessarily expect it to be all good geometry. Also, in the predictions, there's very little information about residues that are far apart, because the multiple sequence alignment doesn't give you any information on that. And errors in, small errors in distances between residues can propagate through the structure. So the models may end up having distortions and also incorrect domain relationships. These are both observed. So here's an example of limitations. Here's a little piece of a structure predicted by AlphaFold, where there's a structure that this deposited structure that goes with it, 6TE3, and Tristan Kroll rebuilt this model and gave me this slide that he got from that. So the blue mesh is the density from the crystal structure, and the blue model is the AlphaFold model. And you'll notice that the AlphaFold model is pretty close, but it doesn't exactly fit the density. And the side chain at this position on the upper left, turns out it's a pretty uncommon rotamer that was put in. So that's because AlphaFold doesn't know, is not perfect in generating the right geometries. This was a high confidence residue. It also has a poor fit to density. Of course, AlphaFold didn't know anything about the density map. So Tristan rebuilt that and put in a more common side chain, and that fits the density very well. So once again, AlphaFold models turn out to be great hypotheses, but they're not perfect. Let me emphasize also about local accuracy is better than global accuracy. So here's going to be a prediction of a large ring structure, 7BGL, has 26 copies in the chain. And so we predict this with AlphaFold. And what do we get? Well, doesn't look all that good. There's huge overlaps. But then if we look in a little bit more detail and ask, what about one of the chains? Well, here's one of those chains, and yeah, it still has that bad overlap, but now let's compare it with one chain from the deposited structure, and that's actually pretty good. So the local accuracy can often be much better than global. So if you're using these models, you want to keep those things in mind. And once again, they're great hypotheses. So to summarize that part, AlphaFold models are great hypotheses. Parts of them are very accurate. Parts are completely wrong. The confidence measure is helpful, but may not be fully reflective of accuracy. So these are very useful characteristics for using AlphaFold or Rosetta Fold or other models like this in any context. So now let's look at this in the context of structure determination by X-ray and cryo-EM. How can we use these features of AlphaFold models? So one thing we can do is, as I mentioned earlier, we can use the confidence measure to prune what's likely to be the worst part of the models away. And then the remaining parts may be pretty accurate. And it's important to notice, this is a little subtle, this is better than a standard homology model because the AlphaFold models almost always do not have insertions or deletions in the sequence. So if you have a structure in the PDB and you have your sequence and the sequences are a little bit different and one might have insertions relative to the other, it may be difficult to get the sequence alignment just perfect in your homology model. The AlphaFold models don't have that problem. So that means that the pieces that are built are almost always correct in sequence register. So here's an example of what you can do today with an AlphaFold model. And if you get into a little bit of a problem with solving a structure, this is a great way to go anytime. So this is a real example from Jamie Wallen. He sent this data to us actually a year and a half ago, and we were unable to help him at that time. After AlphaFold came out, we were able to help him. So this is a crystal structure, and this is the model he had at that time, had a RR free that was a little bit high for the resolution, and he didn't have parts of it built. And here's why. There's the map that went with this, and it just isn't obvious how to connect residues 118 to 139 and to 128 and 139 to 147. It's just not clear where it goes. So if you send me this today, what am I going to do? I'm going to take the sequence, I'm going to generate an AlphaFold model, and then I'm going to just superimpose that model on the model you're working with. And here's the result of doing that. Wow, this is just amazing. Most of the AlphaFold model, which didn't know anything about this model, superimposes very closely, and then it generates for us a great hypothesis about how to connect 118 to 128 and 139 to 147. And so now we can test that hypothesis. That's not very hard. We just look at the density map and see what it looks like. And wow, this is a great result. It's a great hypothesis. It fits the density map as well as you could possibly imagine at this resolution and with this quality map. So this was a really nice use of AlphaFold. And after refinement of that structure, starting with that AlphaFold model, the RR-free decrease as hoped for a structure at this resolution. So it was, in fact, a very good hypothesis. So that's a very simple use of AlphaFold. And just keep in mind, AlphaFold is a great way to generate hypotheses. Just briefly, others have used AlphaFold in molecular replacement, and I'm just giving you an example from Jim Hurley's lab where they showed that AlphaFold for this particular structure had generated a model that was partly the same as the right answer and partly a little different. Molecular replacement worked pretty well at 2 angstroms, and some parts were very accurate, and some parts were rather wrong, and had to be rebuilt, such as side chains. And parts of the main chain required actual rebuilding. So this is a typical situation. The AlphaFold is going to generate some things that are right and some things not. As a cryo-EM example, let's come back to the structure I've showed you a few times. This is 7MJS, and I showed you this picture already that part of the structure was not that accurate. So what can we do? Well, let's trim the low confidence regions. So we're not trimming the bad regions, we're trimming the parts that AlphaFold has low confidence in the prediction, which happens to be the bad regions in this case. And then we can take the density map and we can dock that high confidence remaining part into the map. And then the part that was missing, we can use the density map to trace the chain. And that generates a completed model, which is not that great in the part that was traced, but actually now it's much closer to the right answer than it was before. Okay, so now the part that's quite new, you can actually improve this whole modeling process in an iterative way by using information from a density map. It can be from cryo-EM, can be from X-ray. So let's see how that works. This is the picture I just showed you of the rebuilt model, that a model that was rebuilt using a density map starting from the AlphaFold prediction of 7MJS. And it looks something like the right answer, but it's not perfect. So let's ask the question, can we take this rebuilt model and can we get a better AlphaFold model if we use that in the prediction process? So let's go back to that prediction procedure that we talked about. So in the usual case, we're going to start with a sequence and a multiple sequence alignment, all the parameters, and we're going to generate a prediction, which, as we saw in this case, was not so good in this region of residues 100 to 120. So now, instead, let's add in another piece of information, a template. And so what's different here? So the multiple sequence alignment gave us information about what residues are close to what other residues. And that's the main purpose, the main piece of information coming from the MSA. Now we have a template. The template also gives us that information, actually gives us several pieces of information. It gives us suggestions for backbone angles, and it also allows AlphaFold to focus attention on residues that actually are close. So it tells AlphaFold which residue pairs to be thinking about when it's doing the prediction. This is important because AlphaFold is not able to search all the possibilities for this confirmation of a structure. What it does is it focuses attention on residues, groups of residues that are likely to be close based on that multiple sequence alignment or based on a template, and then basically folds those parts and then groups those at higher level and higher level until it builds up the whole thing. So it isn't able to do everything all at once. So focusing attention on the residues that are close is very important. So now using that template to focus attention on the right residues, now it's able to do a better three-dimensional prediction. So that means, yes, the prediction is better. And the amazing thing is the new prediction is better than the template. So the upper right in green, you can just see it. It looks a lot better than that template, and it's actually much closer to the right answer. We'll see that later. So this is really powerful. You can make a prediction that's better than a template. That means you might be able to iterate the whole process. Here's another example of prediction with a template, just to show you the power of this concept of having a template that generally shows AlphaFold what residues are close, and then AlphaFold can make that better. So you can think of this next example as kind of refinement with AlphaFold. So this is actually that same structure I showed you before from Jamie Wallen. It's a phage repressor complex, and this is just a little teeny piece of the structure showing that the model that Jamie had a year and a half ago didn't quite fit the density right in this particular region. And you can see that a few residues don't match very well. And now we're going to do a prediction with AlphaFold that's a little unusual. We're just going to take this refined model as a template. We're going to run AlphaFold with no multiple sequence alignment and generate a new AlphaFold model. What happens in this case is the new AlphaFold model actually changes the register in this region. So now those residues that didn't match the density actually match the density pretty well. So this shows the power of the AlphaFold prediction with a template. It doesn't just use the template and take exactly what the template says. It uses the template as a guide for what is close to what, and then it does a prediction of a structure that is compatible with the sequence. So this, as I said, you can think of as refinement with AlphaFold. Very powerful possibilities. So now let's do this in an iterative way. Let's take the structure 7MJS that was predicted by AlphaFold and compare it with the map, and then we're looking at that here. On the left-hand side is kind of the good part of that prediction. The right-hand side is where it was a bad prediction. And then, as I showed you, we're going to rebuild that model. And the rebuilt model is not that great in this particular case. Actually, it has a lot of bad geometry. We're just going to feed that rebuilt model in as a template to AlphaFold. And AlphaFold is going to generate a new model that's better than the one it did before. This doesn't quite fit the map yet. But this model is so much better that it can be rebuilt very easily. And that rebuilt model now fits the density. Now we feed that one back into AlphaFold. And that AlphaFold model now fits the density quite well. Notice these AlphaFold models that I'm superimposing here, they're not refined. These are just the AlphaFold model as is and just docked into the map. And we iterate this a couple times, and now we finally have an AlphaFold model that, in fact, matches the deposited structure for this crystal for this cryo-EM map very closely. And as I said, the model is just superimposed here. So we've actually changed the prediction using this information from the map in an implicit way by generating a new model, using that model as a template, and then the new AlphaFold prediction using that is much closer to both the map, and it's compatible with geometric criteria that that AlphaFold has. So this is a really powerful thing. So you rebuild the AlphaFold model with a density map. That rebuilt model improves the next AlphaFold prediction, and you iterate to improve the model. So you can think of this in either of two ways. One way to think of this is how to make an AlphaFold prediction that's better. So you can stop at this point after you've generated a new AlphaFold model, and now this one's a better AlphaFold model than the first cycle. Second way to think about the same thing is you can take that AlphaFold model and then rebuild it into the map. And so each cycle you're getting a rebuilt model that's better. And either way is fine to think of this. The nice way of reason to think of it the first way as this is a procedure for generating good AlphaFold models is that AlphaFold models, despite their limitations, often have very good geometry. So this is a great way to generate a model with good geometry that also is more or less compatible with a map. Okay, so finally now, what can we do with all this in terms of structure determination with cryo-EM and X-ray? How should we think about solving a structure today given all this power of the AlphaFold prediction procedures. Well, here's a strategy for doing this. First thing you're going to do, and I think the first thing you should do starting now, is get a starting prediction for every chain that can be modeled. So you have a biological question, and you have a molecule that you would like to know, what does this look like? So the very first thing you should do is generate an AlphaFold. If you don't already have it, generate an AlphaFold model for every chain, or as for a group of chains, if you can do it that way. And what does that AlphaFold model tell you? That AlphaFold model right away is going to come with that prediction of confidence. So then you can trim that AlphaFold model and get rid of the stuff where it's low confidence. So then you know what you know something about and what you don't know anything about. So this tells you right away, I know a lot about this part of the molecule, and I don't know much about that part of the molecule. And so you already have a great hypothesis for a lot of your structure. And if you're lucky, you'll have a lot of it there. And if you're not lucky, you won't have much. And so now you know something that you didn't know a few minutes before. And this doesn't take all that long. So generating a AlphaFold prediction for a totally new structure, if you have, let's say, 200 residues, that's like 10 minutes. And if it's 1,000 residues, okay, maybe it's an hour. But it's not very long. So you can generate the whole thing, in a day. You'll be all done with that. And you can really start thinking about your structure before you do your experiment. So now you have some information about, what does my experiment have to tell me to answer the actual question that I'm interested in? So now you can think about that, and now you can design your experiment, and you can decide what method you're going to use to try to get experimental information. Okay, suppose now you do all that, and now you spent months, right, generating your sample, and you've got your cryo-EM map, or you've got an X-ray data, or whatever. And so now what are you going to do? So now you're going to take those predictions, you're going to use them. So the first thing we're going to do, so you're going to get a density map one way or another. And so if you have cryo-EM, you've already got it. In X-ray, you're going to do molecular replacement probably with those predictions. We'll talk more about that later if you want. And then, so now you've got a map. So now we can redo those predictions, just like we just saw. You can use the map to improve or get a chain for each chain in the model. So if your AlphaFold prediction didn't work at all for some chain, maybe your map is good and you can build a model. Maybe it's not that great, but maybe it is. And then, so now you have updated versions of every chain, and you're going to use those updated. Oh, I should just mention, in the old days, you would improve this map iteratively if you had an X-ray structure by iterative model building, density modification. Now we're going to do something a little different. Instead, we're going to take the new model that we just built by rebuilding the model based on the map, and we're going to generate a new prediction using those rebuilt models. So now we have a new prediction that's better than we had before, and we can go back to the density map stage, iterate this whole procedure as many times as you want. And a lot of this can be automated, so you don't necessarily have to do all this by hand. You can do much of this in an automated way already, so you can kind of get through this stage without doing a lot of manual work. That'll be later after this is all done. So what does this sum up to? You're going to predict the chains at the beginning before you do any experiment. You're going to get a map. You're going to place the model in the map, the models in the map. You're going to improve the models by rebuilding them, maybe automatically. You're going to get better predictions, and you'll iterate that process. So what's really changed here? Well, before AlphaFold, you started with a homology model, and then you got your map, and you put the models in the map, and you improved them. If you had X-ray data, you iterated that process, and if you had cryo-EM, you didn't. With AlphaFold, you're going to start with a predicted model, and then you're going to get a new map, and you're going to place the models in the map. And then instead of going back, you're going to go on and get a better prediction, a better model. You iterate that process. So how different is all this? Well, it's different at the first stage. So instead of that homology model, you've got a high accuracy predicted model. So in general, you'll have a much more accurate and much more complete starting model to work with. So that's a really big deal already. Then second, now with either X-ray or cryo-EM data, you can make cycles of improvement of the modeling process, of the model and map process. And I'll maybe mention in a moment about cryo-EM. So you can generate a new map this way for both X-ray and cryo-EM. And third, even in X-ray data, you'll have more improvement each cycle because you're using the new model as a template in prediction to generate a better prediction every cycle. Whereas in the old way with X-ray data, you had this a model that was just built into the new map, but no extra predictions. So you get 3 different levels of improvement with AlphaFold compared to what you had a year ago or a year and a half ago. Just a comment on the cryo-EM data improvement with of a map with a model. There is now density modification of cryo-EM maps available, and you can use a model in that density modification process, so you can improve a cryo-EM map using a model. There is some risk of model bias, but so far actually there's not any evidence of model bias. Okay, so I'm going to do really quickly 2 minutes worth of what difference does all this make. So this figure shows you prediction of a SARS-CoV-2 spike protein complex, actually, but just showing the spike protein here, 7-MLZ, and the brown here is the deposited structure, and the blue is the predicted structure from AlphaFold. And in this case, the predicted structure is pretty close, but not that close. And after iterated rebuilding, it's a lot closer. And I'm going to skip that one. This slide shows, does this make a difference in sort of real life for real structures? So we took 25 recent structures, all determined after AlphaFold database was created. And this is a plot where the points above the line are good, basically. So the x-axis is how many residues in the AlphaFold predicted model are within 2 angstroms of the deposited model after superposition, and the percentage. And the y-axis is the same thing after iterative prediction and rebuilding. So basically, after iterative prediction and rebuilding, a lot more of the residues are close to the right answer. But it's not perfect, but it's a lot better. And skip this one. And let me just jump now to where are we after doing all this? So we have procedures for iterative rebuilding of models, for generating AlphaFold models. And so you do this whole process, you can do it automatically. And then where are you? Well, actually, this is the same as always. Nothing has changed. You still have to identify errors. You have to refine the model, add covalent modification, add ligands, add alternate conformations, add solvent, estimate the uncertainties. All this is just the same as it always was. We have tools in Phoenix to do all these things. So Colab, so Phoenix, Google Colab is a very nice, mostly free tool for doing calculations like this. And so in our Phoenix tools, we have tools to basically load up and send you to Colab to generate AlphaFold models and to improve models with the density map. We have tools in Phoenix also to trim models and identify the good parts and to rebuild models. And finally, I want to just give you just a couple comments on AlphaFold predictions, just for you to think about when you're using AlphaFold or Rosetta Fold for any purposes. So the MSAs, multiple sequence alignments, and the templates tell AlphaFold what's close. That's the main thing that they are doing. And that means if you have a really good MSA, you don't really need templates. So you don't need templates from the PDB, which is kind of an option in AlphaFold, if you have a very good MSA. In contrast, if you have a very good template, you don't need a multiple sequence alignment. You can use both, though. Another thing to keep in mind is that all the AlphaFold inputs basically go in a table that's one column per residue. So the features that go into the prediction are basically features that are keyed by residue. So any incorrect alignment in a multiple sequence alignment will basically put information in the wrong key, in the wrong column, and that will make it worse. So incorrect alignments are very bad. Multiple confirmations that are present in a structure would lead to multiple sequence alignments that may be incompatible with each other and complex. So if you are working with a protein that does have multiple conformations in nature, then the multiple sequence alignments may not be simple to interpret, and it may be worthwhile to develop technologies to separate out those MSAs that correspond to one conformation or one complex that or compared to another. Another thing that is a corollary of this one column per residue is that any template that cannot be aligned to the sequence or not correctly aligned are useless. So if you come in with a template that doesn't have any sequence information that corresponds to the sequence that you're trying to predict, that's a useless template. Okay, so I just want to end there and thank all the members of the Phoenix team that have contributed to this work, and also to the many users who have supplied data and problems and suggestions that make all this possible. Thanks, and I'll be happy to have some discussion.
Thank you, Tom, for this great talk. There's some questions in the chat. James Krieger asked, could the open ring ending up closed in AlphaFold be an example of alternative conformer hypothesis?
Let's see, I'm not quite clear on the question. Maybe, Jose Luis, can you explain what?
You showed in one of your slides earlier on, there was a structure that had multiple subunits that they kind of assembled into some kind of archway, not quite a ring, and then the AlphaFold model, it seems like it's much more closed. Whether this is really something incorrect from AlphaFold or whether it could be some different confirmation of how these subunits are arranging themselves.
Yes, I see. That's a great question. And yeah, I think that the general concept that you're suggesting there of can AlphaFold be suggesting to you a hypothesis that you should be thinking about. So in general, the answer to your question is yes. In this particular case, the answer is no. AlphaFold really just doesn't think about those really long distance contacts, and so it just put everything into a big bundle with overlapping atoms. So basically, the AlphaFold model is impossible in this particular case. But in the general case, I think you're totally right. And using AlphaFold as a method for generating multiple hypotheses is a great idea. And there are lots of ways one might do that. So for example, by supplying multiple sequence alignments that are just a subset of the total. That's one way that AlphaFold actually already does that, and doing that systematically might allow you to generate better hypotheses. Another way to do it is to supply templates that are somewhat varied themselves, and then each template will give you a little bit different prediction, and that might be a good way to generate diversity as well. So the general idea is really good, yes.
Thank you.
Well, we have more questions from James. Has AlphaFold learned to overcome incorrect alignments, or are they very good at doing alignments?
That's a great question. I don't really know the answer. So the question, has AlphaFold learned how to get around incorrect alignments? So I suppose the answer is in part yes, because AlphaFold was fed alignments that were all pre-made, and it must have kind of figured out that these aren't always right, because it knows the answer in these cases. Some of the alignments that was fed at the very beginning in the training were also incorrect. So it presumably has figured out that what I'm given as a multiple sequence alignment isn't always right. It presumably did figure that out, but we don't actually know. It's one of the problems with this type of prediction AI, is that you don't know exactly what is being used. And in fact, today, so I told you, I think that the multiple sequencer alignments are being used in this particular way, right? To generate information about what's close. I said that because that information is definitely there in the MSAs. We don't actually know that AlphaFold even used that, but it seems very, very likely. But we don't have a method to say AlphaFold used this to do the prediction.
I do remember there was, I think it was when they presented it at CASP, it was showing some updating of the distance or contact matrices through the generations of the prediction that did seem to be
Yes.
showing these kinds of features.
Yes. So yeah, that's intrinsic to the AlphaFold prediction procedure, is to iterate the process. They typically do it 3 to 5 times of predicting a structure and then using that structure basically as a template in a similar way to feeding the templates in from the beginning. It's actually slightly different. They actually use 2 different procedures for recycling templates, but yes, that is in fact done. You're right. Yes.
Thank you. Next question.
Next
In a way,
one,
AlphaFold, yeah, I think it's
more of a comment than a question. That
go ahead, James. Ask. I think it's better if you ask directly.
Yeah, it was when you were showing this kind of idea that you rebuild from the density and then you get AlphaFold to make a model again based on that. Did it kind of feel similar to if you use Phoenix or RefMac, and yeah, in this kind of way that it's somehow improving the geometry of these things, that kind of combining some kind of restraints into the way that the modeling is happening, even if it's not directly linked up to the density.
Yes, I definitely agree with that. And to me, it's, yeah, this is a reconstitution of the idea that first came with actually ARP-Warp, like more than 20 years ago, of iterating this process of generating a map and using the map to get a model and then recycling that in some way. So this is a generalization of that same idea that was incredibly powerful back then. And so now we just can do it at another level in the prediction, as you say. Yeah, exactly right.
My last question was that it's not necessarily necessary to say, let's start from a sequence in an MSA and try and get a structure from AlphaFold as a starting point. If I have already some other conformation of my protein, maybe would it make sense to kind of feed that in as a starting point into the process to put that into AlphaFold, maybe after some flexible fitting or something like that.
Let's see, let me just clarify. So you're saying maybe you already built a model in some way, or you had one from a previous
From
experiment?
a previous experiment, then maybe you've
Yes.
changed some ligand or some
Yes.
subunit or something. You expect your conformation to be now different.
Yes.
And if you have a density map that you can say, and I can see that some paths are in different places. Where in the pipeline would you put that AlphaFold? That presumably you'd want to somehow adapt your initial model to be a template somewhere along there, or not?
Yeah, so that's a great question. So I would say, yeah, the pipeline that I described is if you don't know anything, but if you come in with, yeah, maybe you solved a structure before and you've noticed it's got a flexible part, and in this map it looks like that flexible part is going a different way. So I guess in that case, what I would do is I would just go directly to start with the model you had from yesterday and dock it into to your current map and just fit it in there, so that it kind of reflects the new map kind of as well as you want. You could either like really build it seriously or just kind of make a bend there at the link at the loop where it's flexible, and then take that thing as you're suggesting, take that model and include that as a template in the AlphaFold prediction, in your first AlphaFold prediction in this case. And you can just supply any template. You can supply one from the PDB, you can supply a PDB ID, or you can upload a model. And so you upload your model and run your AlphaFold prediction with that as a template. And you could do it with or without multiple sequence alignments. And you might try it both ways. And then use that as your starting point. And so that one, the AlphaFold model, hopefully will then take the confirmation that you're looking for there. So some people have tried this and have told me that they have trouble with this. I think in most cases, the problem is that they haven't still included the multiple sequence alignments, and that those are kind of distracting AlphaFold from the confirmation that you're suggesting. So I definitely encourage you to try it at least both ways, or do include a run where you just supply the template and no multiple sequence alignment. So yeah, your idea is great.
Thank you. And the last question comes from Roberto Maravini, and he asked, can you comment on the challenges that AlphaFold users face when working with multimers instead of monomers?
Yeah, so let's think of two different kinds of multimers. One is like the chain that the structure I just showed you, that ring thing, where there was a whole bunch of copies of the same thing. And then other multimers are like multi-protein complexes with all different chains. And so most of the time, if you have one chain and it's multiple copies of the same chain, most of the time those multiple copies are going to be pretty similar. Not always, but in good fraction of cases it's going to work that way. And so you can run AlphaFold with versions of AlphaFold, not our server but everybody else's, allow you to supply multiple chains. And so basically what it does when it runs AlphaFold prediction with multiple chains, all it really does is it adds a bunch of glycine linkers between your chains, and then it runs them as a single chain. So you can do that yourself if you want to do that. Basically that allows AlphaFold to kind of ignore the linkers there. And that works reasonably well. And in some cases, those models are better. So in fact, the example that I showed you with that ring, depending on how many subunits you supply, you can get a better or worse multimer structure. And if you, in that particular case, if you supply about 5, turns out that gives you actually a very good 5-mer for that one. And then you could build that up to get the whole thing. So part of my answer here is you might, if you have a multimer structure, you might want to try 1, 2, 3, 5, 10, or whatever the number is. Try a few different possibilities, run them all in AlphaFold prediction, and you can just look at them and you'll see which ones are kind of plausible and which ones aren't. So treat them all as hypotheses and evaluate those hypotheses against what you already know and against your data. So that's the simple answer for that one. Similarly, actually, for a multimer, other people have servers where they do multimer prediction. The AlphaFold people also have a new version of AlphaFold, which I don't think anybody uses their server yet. But anyways, it's available to do predictions with. It has a new training there, so you can do predictions with the multimers. We don't have the that capability on in our AlphaFold prediction things, but you could just use any server to generate a multimer prediction. And you asked about challenges. Well, basically the challenges are that the multimer prediction doesn't work all that well. It works pretty well, but you don't get the domain or chain interactions all that accurately in many cases. So you may need to do prediction. If you have experimental data, you're way better off, of course, right? Because you can, if you have cryo-EM data, you can just cut out the density around each chain in your map and do this iterative, and you can generate an AlphaFold model for each chain, basically dock them in, cut out the density around that model, iteratively rebuild it with the tools that are already there, and you can do them one by one and get the answer. If for X-ray, it's a little bit harder because you basically, you need to do molecular placement, and if you have a big structure with many chains, it may be difficult to do molecular placement to get the first chains placed. So my suggestion in that case is, well, a lot of these cases you actually know something. Like if maybe you have a ribosome and that's your case. Well, you already know a lot about what ribosomes are going to look like. You can dock your AlphaFold chains into a ribosome structure that's the homologous one. Use pieces of that as a starting material to try to get a molecular placement solution. So basically, for X-ray, you want to have the biggest piece you can that's as accurate as possible to start off molecular replacement. And so it'll be a little bit of a trial and error thing. If you have a big chain that's a good part of your molecule, you're going to start with that. If you need to do multiple chains, you'll do prediction with multiple chains. Try to get the one that looks like it has the highest confidence and use that as your starting position molecule, a starting search model for molecular replacement. So it's a little harder for crystallography than for cryo-EM, but both I think are pretty doable now in most cases.
Thank you, Tom. I think there's no more questions. I would like to ask you something, but I'm not an expert in models, so I prefer to keep my questions by myself. But at least for people like myself, that we are more focused on the image and cryo-EM, this probably all of us gave us a lot of ideas. Just this opens a lot of possibilities, like perhaps starting with an AlphaFold model, converting into density map for initial volumes and try to avoid local minima.
I think that is a great idea.
Yeah.
Yes. Yes.
And this is the kind of ideas that came to my mind from an image processing point of view. Yes.
I think it's a really good idea. And you could even generalize that as from the point of view of here are AlphaFold models. These are hypotheses for what's in my picture. And then now you have and then you're going to generate a picture. And as you're starting from basically nothing, but just from your data. So your picture should be compatible with at least some of the things that are hypotheses. And though any picture that is compatible with something that's your hypothesis is more probable than a picture that doesn't have anything to do with any model that you've ever seen. So I think that general idea is really great. Yes.
Thank you. And that's my comment. And if there's no more questions, I think we end here. Just thank you, Tom, for this talk and for accepting be with us today.
Okay, thank you very much.